Incubators are specialized devices used to create and maintain controlled environments. These devices ensure stability in temperature, humidity, and gas concentration. Scientists and researchers rely on incubators to grow and preserve cultures, cells, and tissues. The primary purpose of an incubator is to provide a constant and controlled internal environment. This stability is critical for the growth and maintenance of biological samples. Without such precise conditions, it would be challenging to conduct many scientific experiments and medical procedures. In the world of science, maintaining specific conditions is crucial. Even slight deviations in temperature or humidity can affect the results. Incubators help eliminate these variables by providing a stable environment. Understanding how incubators work is essential for anyone involved in scientific research. This essay will delve into the science behind these devices, their types, uses, and maintenance. To maintain a stable environment, incubators control temperature, humidity, and gas concentration. Inside an incubator, heating elements and sensors work together to keep the conditions optimal. These elements ensure that the temperature remains constant, even when the external environment changes. Humidity control is equally important. Most incubators have water reservoirs or humidifying systems to maintain the required moisture levels. This is particularly crucial for certain types of cell and tissue cultures. Gas concentration is another key factor. Some incubators can control the levels of gases like carbon dioxide and oxygen. This ability is vital for growing specific types of cells that require precise gas environments. There are two main types of incubators, hot and cold. Hot incubators are used to grow and maintain cultures at warmer temperatures. Cold incubators, on the other hand, keep samples at lower temperatures, often for preservation purposes. Each type has specific components and features designed to maintain the required conditions. There are 4 common types of incubators used in laboratories: simple, anaerobic, carbon dioxide, and cell culture. Each type serves a unique purpose and provides specific conditions for different scientific needs. Simple incubators are the most basic type. They maintain a constant temperature and are often used for general purposes. These incubators are ideal for growing bacterial cultures that do not require special gas environments. Anaerobic incubators create an oxygen-free environment. They are used for growing and maintaining anaerobic bacteria, which cannot survive in the presence of oxygen. These incubators have gas-tight seals and use nitrogen or other gases to replace oxygen. Carbon dioxide incubators are designed to maintain a specific concentration of carbon dioxide. This is essential for cell culture, as many cells require a precise carbon dioxide level for optimal growth. These incubators also control temperature and humidity, making them versatile for various applications. Cell culture incubators are specialized devices that provide the ideal conditions for growing and maintaining cell cultures. They often have advanced features like HEPA filters to ensure a sterile environment. These incubators are crucial for medical research and biotechnology applications. Section 4. Uses. Incubators have a wide range of uses in laboratories. One of their primary applications is in microbiology. Bacterial cultures are often grown in incubators to study their behavior, test antibiotics, and conduct various experiments. In cell biology, incubators are used to grow and maintain cell cultures. This is essential for studying cellular processes, drug development, and cancer research. The precise control of conditions ensures that cells can thrive and be observed under optimal circumstances. Tissue culture is another important use of incubators. Researchers grow tissues to study their structure and function. This research can lead to breakthroughs in regenerative medicine and organ transplantation. Incubators are also used in IVF or in vitro fertilization clinics. They provide the necessary conditions for fertilizing and growing embryos outside the body. This application has revolutionized reproductive medicine and helped countless couples achieve their dream of having children. Section 5. Precautions and Maintenance. To ensure incubators work effectively, regular maintenance is crucial. One of the most important steps is cleaning. Incubators should be cleaned regularly to prevent contamination. This involves wiping down surfaces and cleaning water reservoirs. Calibration is also essential. Sensors and controls must be checked and adjusted to ensure they are providing accurate readings. This helps maintain the stability of the internal environment. Another key maintenance step is to check for wear and tear. Components like seals and heating elements can degrade over time. Regular inspections can identify issues before they lead to failures. Proper usage is just as important as maintenance. Users should follow manufacturer guidelines and avoid overloading the incubator. This ensures that the device can maintain the required conditions effectively. Section 6. Conclusion. Incubators play a vital role in scientific research and medical applications. They provide stable environments essential for the growth and maintenance of cultures, cells, and tissues. Without incubators, many scientific advancements would not be possible. Regular maintenance and careful use are crucial to ensure incubators function optimally. This involves cleaning, calibration, and inspections. By following these steps, researchers can rely on incubators for accurate and consistent results. In summary, incubators are indispensable tools in the scientific community. Their ability to create and maintain controlled environments has paved the way for numerous discoveries and innovations. Proper care and maintenance will ensure they continue to support scientific progress for years to come.